Hi, so we're now going to process our data that we gathered using 5 different flasks, 5 different standard flasks that were filled with dichromate and acid and a lot of water and a small amount of alcohol, depending on which flask it was. Once we had reacted those, mixed them with water, and then measured them at 580 nanometers, we were able to figure out the relationship between the percent alcohol, or the concentration, versus absorbance. Remember, the reason we chose 580 nanometers was it was a relative maximum for the Cr3+. This chromium 3 plus was a direct result of the reaction of our dichromate, our acidified dichromate solution, and ethanol. So when we just had the acidified dichromate, there was no presence of chromium 3 plus at all. As soon as we added the alcohol and let it react, the concentration of chromium 3 plus increased until it reached equilibrium, and that blue-green we could measure at 580 nanometers. So this was the data that I got when I did this experiment. My 0% alcohol was my blank, and I kept that for use throughout the experiments. So my first one was a blank, and I set my absorbance to read 0 with this particular sample. Then I measured the contents of flask 1 at 580 nanometers, and I got an absorbance of 0.116. The third flask, which contained a half a milliliter of 10% alcohol, I measured at 0.265. The fourth, which contained 0.8 milliliters, I measured at 0.468. And number 5, which had 1 milliliter of alcohol, I measured at 0.634. Now, the 0.2 milliliters, remember, corresponded to what we labeled our graph as 2% on the concentration. 0.5 milliliters we called 5%. 0.8 milliliters we called 8%, and 1 milliliter we called 10%. So we plotted absorbance on the y-axis and percent alcohol on the x. And when I did this with mine, this was the graph I got. I had the two variables. One was independent and one was dependent. Concentration is the independent variable. We controlled the concentration. We made those solutions, and then we put them in the spectrometer, and the result of that solution gave us our dependent variable. Which is the absorbance. Dependent. It depends on the concentration and the solution that we made. So the dependent variable is absorbance, the independent variable is concentration, and we plotted the independent variable on the x-axis, dependent variable on the y-axis, and we came up with, or I came up with, this equation of a line. Y equals 0.0621 x minus 0.0141. This, remember, is sort of our error. So absorbance equals our slope, this is our molar absorptivity, times the concentration, that was x, minus that little error, 0.0141. Now we went on to the second part of our lab, and in my case, I measured a mouthwash, 2 different samples I made, and a beer. Again, I made 2 different samples. Now, these samples were supposed to be exactly identical, so I should have had absorbances that were exactly identical. Well, they weren't too bad, and what I did was take this absorbance, just for an example, And I plugged that into that equation. Remember, we had the absorbance equals 0.0621 minus 0.0141. So if I had the absorbance, which I now do, I could plug it right in and solve for the percent alcohol. But let's rearrange this equation so it makes it easier. First of all, we're going to add that same 0.0141 to both sides. That cancels this. So we get absorbance plus 0.0141 equals 0.0621 times our percent alcohol. The next thing, of course, we do is divide both sides by 0.0621. And that takes care of this over here. So now we have absorbance plus 0.0141, the whole thing divided by 0.0621 equals our percent alcohol. So when I took my 0.272 and plugged it in here, and then added 0.0141 to it, and then divided that whole thing by 0.0621, I got, 4.61% alcohol. Okay, that's all I did. I used this equation. If that's true, what's going on here? Well, mouthwash actually has about between 1/4 and 1/3% alcohol in it. Okay, it's a lot of alcohol in mouthwash. So in order to be able to get it to read in our spectrophotometer, or Mr. Proetta diluted this by 5. So once you find the percent alcohol in your diluted mouthwash sample, you multiply it by 5 to get the actual percent alcohol. And we did that in both cases here and found that our alcohol was 28-29% for the Listerine. The Busch Beer, on the other hand, was between 4.6 and 4.8, and we did not have to dilute that because it was low enough in alcohol.